appreciate the effort I put into the title. I really wanted to fit into the theme of the workshop. Okay, so as I understand, there are some students here who are computer science students who is trying to get a feeler for the field. So I'll spend a little more time on the motivation than I would have otherwise. So my talk will be all about transcriptional regulation and why do we care. So the big $1 million reason why we are doing what we are doing is this link here from genotype to phenotype, right? How differences in our genome manifest themselves in differences in phenotypes. And most of us will agree that this link from genotype to phenotype in large part goes through this thing called transcriptome. So the genetic differences lead to difference in transcriptome, which in turn, through crazy nonlinear relationships here, manifest themselves as phenotypes differences. And at all levels, the diversity in nature, the underlying cause is transcriptional regulation. So that's why we care about this process. To set up the scene of what all goes on, transcriptional regulation, how genes are turned on in specific conditions. There are lots of things goes on, but I just want you to focus right now on specific thing called interaction between these transcription factors to the DNA. And that happens in many different places in the genome, specifically near the gene, where gene starts at the promoter. But more recently, we know that it happens very critically, very far away from the genes as well, especially for the genes that are highly condition specific. So my focus will be a lot on this part here. So just to zoom in a little bit on distal enhancers, this is a poster child of when we talk about distal enhancers. This is a sonic hedgehog gene, and its enhancer is located about 1 megabase away in the intron of some other gene. How would we ever find this out? That's crazy, right? And single nucleotide mutations here can result in extra, fingers. It's that important. Okay, so with that quick background, there are 3 quick stories I will.
Just a question. So everybody always presents this Sony hedgehog example. Are there other examples?
There are many other examples. So everybody presents this one because this is the farthest enhancer known. And there are lots and lots of experimental data in many, many mammalian species where polymorphisms in this enhancer have been shown to result in deformation. So that's the reason. But there are many other examples, not as extreme as this. And the phenotypic consequences is not as extreme as this. But there are many examples for sure, at this stage for sure. Okay, so the first part of the talk will be this relationship between SNPs and gene expression, and I can turn it around and say the same thing: relationship between enhancers and genes. So that takes us to this field called genome-wide association studies. So what is it? So it's basically you have 2 groups of people, affected and unaffected, for some phenotype of interest, and we are looking at one polymorphism at a time and asking whether there is a very differential allele frequencies between the 2 groups, and if they do, then we have somewhere to go in terms of looking for cause of this disease or phenotype. Okay. If my phenotype happens to be gene expression for a specific gene, then this whole thing is called eQTL, expression quantitative trait loci. So same thing, you're looking at one SNP here, and we are asking, given the genotype at that SNP, are the expression levels very different in individuals who have this genotype versus this genotype versus this genotype? Okay? So that's expression eQTL for you. So the big problem with this whole GWAS and eQTL, any kind of large-scale association study, is that that's all they are. They're associations, right? Not causality. And big part of that reason is this thing called linkage disequilibrium. So there are large regions of the genome that through evolution have traveled together as a molecule. So all the polymorphisms in this piece of DNA are linked. So they don't give you any additional information. So you cannot distinguish one from the other when it comes down to causality, because they have exactly the same association with the whatever phenotype we are looking at. So that's one big problem. And second problem is, even when you find these signals, it's very difficult to go and interpret them exactly how they affect the phenotype or gene expression. But unlike general phenotypes, in case of when our phenotype is gene expression, we know a lot about the mechanism of transcriptional regulation. So the idea behind this project is to bring in everything we know to do a better job at dealing with these two major problems. Okay? Because functional interpretation is built in the model itself. So anything we are detecting will have a specific interpretation to begin with. So the basic idea in a nutshell is this. So we have lots of SNPs that may be associated with the expression of a gene, okay? And the goal is to distinguish among them which is more likely to be the causal polymorphism. Okay? And we bring in all kinds of additional biological information grounded in the mechanistic understanding of this process. So there are two classes of information we bring in the model. One is how likely are these individual SNPs to be a regulator, i.e. distal enhancer. Okay? So we have lots of epigenomic data now that tell us specific combinations of them correspond to enhancer activity. So we bring in all this data. This is one kind of information. Even when it is an enhancer, who is to say that it actually regulates that gene and not some other gene? So now we have this, we have to bring in another kind of feature set which link this location to this gene, right? So we have, you can think of 3D interaction. You can think about the correlation between what happens at this loci across conditions and what happens to this gene expression. The two kinds of interaction features that we bring in. We put all this into a Bayesian, hierarchical Bayesian framework and try to solve it. There are more technical tricks that I am happy to admit that my student knows much more than I do, so but I will defer to him. Okay, there's more technical details. So it's basically coupled Bayesian model. So knowing the model for enhancers, which based on all this epigenomic data, helps us home in on specific SNPs, and knowing which SNPs are regulators helps us refine this model. So there is a back and forth between these two models, and it converges. So quick application. So data we had access to were 300 human heart from both failed heart and healthy heart and all that, but we are not making that distinction right now. And we had genotype and gene expression. And we got all kinds of histone modification and other kind of epigenomic data from ENCODE for heart-specific tissues and cell types. So statistics first, then we'll come to biology. So in terms of how much of expression variance is explained by the model, okay? So we are comparing our method with few other methods. I'm only going to show here two representative. One is just a standard multi-regression, Lasso, and one is a tool from Jonathan Pritchard's group called eQTL minor. That is the closest to what we are doing. It does take into account prior that depends on the epigenomic signals. So that's how I'm comparing with this. So in terms of just the explained variance, we see that there is a big difference in terms of how much of the variance is explained. And in this two plots here, what we are trying to, the message here is that all the SNP arrays, SNPs we look at, are a tiny fraction of all the polymorphisms, right? These are tagged SNPs. So typically people impute the SNPs. So we have 10 times more SNPs now. But the people have made this point that you have to look at imputed set of SNPs to really get to the causal ones, right? So when you impute them, you have even a bigger problem. The haystack is even bigger to find the needle in. And that's when power of these methods where you are using the known mechanism comes into play very strongly. Now to the biology. So how do we know? Yes, please.
Can you go back to the method and pinpoint what makes it different from the other?
Right. So, okay, the two main things. One is, okay, so just to make the contrast between this eQTL and minor, what it does is it relies on a prior model of what enhancer looks like. Okay, so for each gene, it looks at the strongest associated SNP. Okay, within 200 kb or something of the gene. That's one limitation right away. Using the strongest SNP for every gene, it builds an epigenomic-based model of what enhancer should look like. So there is no back and forth. And it applies that model and broadens the scope, and then fits the Bayesian model again. What we are doing is, number one, we are looking at a much broader region. We are not relying on a specific model of enhancer. We are letting the model itself be heterogeneous through MCMC sampling. And so, and also the way we are doing sampling takes very specific care of LD blocks. We are making sure that representatives from each LD blocks are fairly sampled. So those are the three main reasons why we think we are doing better. And in the paper, we have tried to make this argument individually.
But are you using the information on known enhancer locations? Or are you doing it for everything within this megabase?
We are everything within this mega, all SNPs, at all SNP locations, right?
So the fact that you call it enhancer is an afterthought. I mean,
Afterthought, but that's right. That's a good point. That's a good point. But that's a good point, yeah.
But how does the epigenetics and interaction data go into your model?
So there is a model for, let me see if there is a picture here. Right. So there is a enhancer gene interaction model, right? So given the location of the enhancer, the SNP, and the gene, so I can say, okay, what is the correlation between DNA's hypersensitivity, for example, at this location across many, many different cell types, and the gene expression, right? So these are kinds of things that go in the model. Okay, so the first thing we asked was the SNPs we find versus SNPs you would find otherwise. Is there a clear difference in terms of evidence of protein binding, right? So there is something called DNA's footprint data. These are high resolution, high depth DNA experiments where you can look at indentations in the signal. Any indentation indicates binding of some protein. So just as a basic assessment, what fraction of what we are calling a SNP show this indentation, okay, show something binding there. So that's number one. So these are, we did that in many, many cell types. And so the first one, for example, we take the union of all heart-related tissue for which this footprint data is available, and we ask what fraction of what we are calling a SNP. And by the way, we control for the number of SNPs we are picking, obviously. It's like, most of the things what we are detecting, even though we don't use footprint itself in the model at all, we are using other indications of the enhancer-wide signals, but at that location, specific location, we are seeing greater fraction of indentations, okay? Evidence of protein binding.
But could it be that in your model you're using something that is highly correlated with the DNA's footprinting?
Yeah, nothing in our model is at that resolution at all. Everything in our model is the 500 base pair resolution, but here we are assessing at that 20 base pair or 10 base pair, 5 base pair. Yeah.
So just following on that, isn't it that in the open chromatin overall you have more SNPs? I think that's probably the case.
In enhancers?
No,
in
open chromatin.
In open chromatin, yeah.
So that probably make it more likely.
That's right, that's true. But the thing is the eQTL minor also is looking at the open chromatin only. Yeah, so control is there. So this is motif disruption. What I mean by that, so we ask for each known binding motif that we are aware of. We ask, and for a given SNP, we can calculate the difference in binding energy for SNP A versus SNP B. And if that binding energy difference is too much, above a certain threshold, we call it disrupted. So for each transcription factor, we ask how much is it disrupted genome-wide by the SNPs we are detecting. Okay? And we rank all the transcription factors based on that. And I'm just showing the rank list here. So this is basically all the red ones are known core cardiac transcription factors. And I did some literature search, and all the blue ones, there are clear indications of their involvement in gene regulation in cardiomyocytes or heart tissue. So this checks out nicely. And then getting to more specifics, like if we are claiming that these SNPs are causal, then we better see a allelic effect, right? So we are just looking at the DNA's hypersensitivity in that region and asking how different are these signals for the two alleles. Okay? We are just calculating the allelic difference, so that checks out. And lastly, we are, in this particular run of the model, we did not use the 3D interaction data directly. We did not use the ChiaPad or 3C or whatever. So here we are actually asking, what is the evidence of 3D interaction between the SNP we are calling and its purported target gene? Okay, so that was quick story number one. Now I'm going to move on to enhancer-enhancer interaction. So this work was done a few years ago already. And the idea was very simple. So we have been talking about gene co-expression regulatory networks and these kinds of things where you look at two genes and see how they go up and down across conditions or tissues, and you do correlation and you have a network. So the data had just become available, so it made sense to just do that for enhancers, right? So like in gene, we are calculating the expression of the gene across conditions. How do we proxy enhancer activity? So at that time, the simplest thing we could do was just look at the open-close status, DNA's hypersensitivity of this enhancer across conditions or cell types. Okay? So just based on that simple idea, we looked at the correlation, but the only methodological trick we had to do was very carefully control for this propensity of things that are nearby in the genome to be nearby in space as well. So that overwhelms the signal, as you can imagine. So we very carefully accounted for that, and then we applied this very stringent FDR and all that. This is just an example here. So each line here is an enhancer throughout the genome. It's a very large region, a couple of hundred megabases. And in these 3 cell types, these are on. In these 2 cell types, they're off. So these enhancers will be part of a highly correlated cluster, for example. Okay. So we apply all these controls and stuff, and we find highly correlated enhancer pairs and clusters, and we look at what are the properties of these things, because that leads into my next project. So these are the main things we found. So first of all, the first question was, do they share anything, the things that are highly correlated? And yes, they do. But more interesting thing is the second point here. Okay, so the enhancers that are very far on the genome and enhancers that are close by relatively, the one that are far have much greater transcription factor binding site sharing. So there is a very specific mechanism apparently for things that are very far and come together in space or whatever.
Could it be simply because you didn't control completely for the distance in the genome?
How would that lead to this result?
Because your correlations for nearby enhancers are not as trustworthy as correlations for far enhancers.
But by the way, when I say nearby here, I'm not talking about the nearby that you would do without the control. The nearby in that context would be, 50 kb, 20 kb, that kind of thing. These are still megabase away versus 10 megabase away. That kind of distance resolution we're talking about here. But yeah, there's something to look into for sure. Other thing we found is that the binding sites that are preferentially shared between these correlated enhancers interact with very specific chromatin modification enzyme. So that was another question, right? How are they impacting correlated activity of enhancers? And one obvious thought was, well, maybe they interact with chromatin modification and change the status of all these things that are nearby. Okay. And lastly, we again checked these correlated enhancers tend to be together in space. So at that time, the 3C data or high C data was available only for 2 tissues. So we just, based on those, there's a big differential in 3D proximity of this correlated enhancer versus uncorrelated. Okay. Just an example of enhancer and gene clusters. So what the model we are thinking in terms of, so the bottom line for the cell's functioning is this correlated activity of genes. How it is achieved is what we are after right now. Okay, so the thought here is that one direct route would be if you had, in the regulatory regions of these genes, if you have shared transcription factor binding sites, that would be easiest way to make their activity correlated, right? That's one. But what we are also thinking is that maybe there is the link between this and this is indirect. Maybe motif sharing somehow affects the 3D proximity that may affect directly or indirectly here the enhancer activity correlation, and that would, of course, directly affect their target gene correlated activity. So this is the model we are thinking about. Okay, so I have time.
Can I ask you a question?
Please.
So you can draw a network of gene expression correlation, right, that is based on motif sharing, and you can draw one that is more careful and based on your knowledge of enhancers and how they impact expression. How different are these networks?
Direct gene correlation expression network and enhancer network. Enhancer-based gene expression network.
No.
Yeah. Okay.
I guess I'm saying how much contribution do we
Right.
as opposed to simple promoters?
Promoters, yeah. That's a good question. I have not specifically asked that, like quantifying the contribution of each of these. I don't know. Yeah. If I have time, we can get back to this question. It's slightly tangential. Okay. So the last part is work is done. We are trying to get it published, but it's a little bit, out there. So we'll see what is the audience reaction here. So the people are really interested in what makes a transcription factor bind to a specific region, location in vivo in the cell, right? So there is a distinction between in vitro, what can happen, in vivo, what does happen, right? So if you just put the TF and the DNA in a test tube and shake it, it might bind, right? But it doesn't bind in real life. So what are the additional determinants of that in vivo binding is of great interest. And these are the kinds of things people have gone down answering this question. So the DNA motif itself is, about 20 base pairs. It doesn't have a lot of information content. It just cannot explain the in vivo binding, specific in vivo binding that well, obviously. Other thought people have is, of course, they work together, so there's cooperative binding. So if you look at combinations of these things, then it explains a little bit more specificity. People have looked at general nature of the DNA around the in vivo bound locations versus controlling for the binding motif itself. That's another. And here is local chromatin state, open, close, all these things. And then this is the most interesting for us, the homotypic clusters of binding sites. So these are, same binding site for the same transcription factor occurring in clusters within a very short region, few hundred base pairs. And these are known to be Ovcharenko has shown that these are characteristics of enhancers. It happens a lot. Okay? And then people in the biophysics community have shown that through modeling this thing called TF diffusion, a facilitated diffusion, where it comes and binds one of those places and it just goes along the genome. Before it goes too far, it's right there, it's kind of trapped locally. So that may increase the robustness of binding by simply having multiple binding sites. It has also been shown that from the evolutionary perspective, it's easier to evolve 3 lousy sites than to evolve 1 really strong site. Right? So these are the reasons that goes behind the huge prevalence of these things called homotypic clusters of binding sites. So we are taking this idea a little further now. So what we are proposing is, or speculating is, what happens when you bring these binding sites that are very far in the genome together in space at the same time, right? So in effect, it's doing what HCTs do, but now it's a spatial clustering as opposed to genomic clustering. And the advantage of such a thing would be it could become very condition specific. If you have just a genomic cluster, it's a static thing. You cannot do much with it, right? It will only depend on the TF concentration. But if you have them differently, then you have TF concentration as one of the determinants and the 3D proximity as another dimension. When they come together, it works. When they don't, it does not. So that's the thought process behind this project. So we are simply going to ask, when, after controlling for the binding affinity that describes the in vitro binding affinity, controlling for that, do we see a higher occupancy, in vivo occupancy, when things are close together in space versus when they are not? That's the question. And how do we quantify in vivo occupancy? We do that using this thing again called footprints. So footprints, so just an example, we have all these footprints and we overlay using very stringent criteria these motifs onto these footprints, and if we see that, then we call it occupied by that transcription factor. If we don't, then we don't. Okay? And we are quantifying among all the places where it could bind, how many of them are occupied. So it's a fractional occupancy. That's what we are calculating. And we are comparing that fractional occupancy between the two conditions, when they are closed and when they are not. Okay? So we call this occupancy boost. How much more the occupancy is controlling for all these other confounders. Yeah?
Is that locally or globally?
So both, because we are talking about a 3D cluster of enhancer elements. So we have predefined these clusters. We have ascertained that they're indeed closed in 3 dimensions, and then that's our space. In that space, we look at all the binding sites for one transcription factor, and we calculate how many of them are actually occupied, and we have a control set of same number of enhancers with all other properties exactly matched in terms of chromatin accessibility, in terms of GC content, and all these things. And we ask what fraction of that binding site is occupied when they're not in 3D space together. So that's the comparison. So we are showing in 2 dimensions the effect of lots of binding sites in space on the in vivo occupancy boost. So the first dimension is the enhancer size. How many enhancers are there in the archipelago, we call it, the cluster or archipelago. And here in this dimension, it's the mean number of binding sites per enhancer. Just to show that it's not just because of a lot of dense clusters concentrated in few enhancers. It's truly a distribution across many, many spatially proximal but geometrically distal locations. Okay, so there's a huge increase in occupancy after controlling for all these other things. So almost 210%, which is like twofold, threefold. So 200% will be threefold occupancy increase. So then we also, just to get a little bit more on and just ground the things into real mechanism, we hooked up with Boris Adrian's group at Cambridge, because he's the one who did the facilitated diffusion modeling for homotypic clusters. So we asked them to see if they can extend that model to see the effect of 3-dimensional proximity. Right? So the direct control here is you set up your system and you're directly comparing the occupancy boost when they are very close, when they are not so close, when they are very far. And we are changing things like how many individual sites are there in per enhancer and how many enhancers are together in that space. So the 2 parameters we control, and this is a highly computationally intensive process, hence these numbers here. But nevertheless, no matter how you look at it, there is a huge occupancy boost when things are together compared to when they are not. So the model was extended in a way that in addition to just crawling along the DNA, it can also jump to a nearby site when they're close enough in space. Okay. Great. So now we have this cluster of enhancers. The question then was, are all enhancers in these clusters made the same? Do they have their individual identity in some way? Are they all affected equally? And for that, we try to distinguish between the enhancers that have lots of binding sites for something of which there is a lot of in that cluster versus the ones which don't. So we call these enriched enhancers, and we call this depleted enhancers within the same cluster. And we compare these two in terms of what is their chromatin accessibility, what is their evolutionary conservation, what is the expression of their downstream gene, and we find clear difference specifically for the enhancer subset, which are enriched in these highly abundant binding sites in the archipelago. Okay? So that's what this is trying to say. So next the question was cause and effect. So it is known in the literature that when lots of TFs bind by whatever means in a region, it affects the overall accessibility of that region. And likewise, other direction is even more obvious. So the question was, what is this chicken and the egg question here, right? So, but fortunately, we can break this cycle by specifically looking at effect of TF gene expression of the transcription factor on the boost. Right? If you see an effect, then that would imply this causality. So that's what this is trying to do. So we divide up all our transcription factors in a particular tissue in terms of highly expressed and less highly expressed, and look at the occupancy boost of the ones that have high expression versus the ones which don't have high expression. And we see this occupancy boost effect only for the TFs that have high expression, not the ones which have low expression. Okay? So that sort of points to that causality. So putting it all together, what we are claiming here is that you can have these HCTs or homotypic clusters of binding sites just lollygagging around in the genome doing nothing. They have to be not only brought together in space like this, but also the specific transcription factor expression has to be there for this effect to take place. And in the same condition, some other parts of the genome where you have everything other than the spatial proximity, the effect will not work. Right? So this is the idea we are. So this kind of really changes the way, I don't know what fraction of control of gene expression this contributes to, but whatever it is, it's like a different way of thinking about adding one more kink to the whole complex mechanistic story of transcription. Like now we have to think about what is nearby in space and why not. So those were the 3 quick stories I talked about, and I'm happy to take questions. And all this work was done by 2 grad students. Now they are postdocs elsewhere. Thank you. Questions?
The last part of your talk, these clusters of enhancers, do you have any idea what they might do to the noise of the gene?
Oh, noise of the gene, okay. That's
very high or very low noise? Would it be very stringent?
If I had to guess, if I had to put money on it, I would guess that will make it robust as opposed to noisy. That would be my guess, but that's excellent. I don't know. I don't know the answer. In fact, just to add on to what we don't know, so people have found this gene and all these enhancers based on various 3D assays. But we don't know the relationship between how these enhancers are controlling the expression of this gene. What is the relationship between what function? What is the underlying function, like mathematical function? So we don't know. Yes, please.
So 2 questions. First of all, how is this related to the classical transcription factories theory?
That's an excellent point. So in our paper, in the discussion, we do say that transcription factories, there are few papers that experimentally look at concentration in specific nuclear areas, and based on that, this theory has evolved, right? But there is no database of transcription factories, right? So our speculation is that this is the mechanism for transcription factory. And we also have in the paper looked at the relationship between this thing called super enhancers, which are high concentration of enhancers in a genomic region. And what we are claiming is that this mechanism sits on top of super enhancer. It can link multiple super enhancers together. So, yeah.
And another question, if you look at the high-C data, most of the contacts are pairwise and not multi-wise. So what's the distribution of the number of
Right. So that was part of our filtering. So we start with just the correlation-based clusters, and we then prune that cluster based on high-C data only to keep the clusters where 80% of all pairs are close by based on this stringent high-C determination. So these...
Typically you will have 2 or typically you will have 10 regions involved?
Oh, in these, what are the size of these enhancers? It can be, in the mean would be in hundreds, lots of dozens of enhancers.
Involved in the
same cluster, highly correlated expression activity, and 80% of all pairs being in high C. Yeah.
Is that geometrically possible to pattern so many? In such as, there is some 3D restriction.
But you can always do the planar thing. It doesn't have to be.
But just to be close in space, the chromatin has some size and all of that.
Right.
So I kind of didn't know is that
Right.
Say hundreds, I start to worry.
Yeah, no, that's a good. That's a good question. So we just looked at high C data and see, okay, are these detected as spatially close based on their FDR? But the distances, I don't know what is the claim on the distance between these two things. And if the distance is too little, then what you're saying is right. It's impossible to have all these things close by. But yeah, these are open questions. I do not know. Yeah.
Okay. We can take more questions in the break. Let's thank the speaker again.
Thank you.